Hello, this is Dr. Jenkins. We are moving right along here, and we are already to chapter 3. Now, this, of course, is going to be about the cell, and the first half of it is very straightforward. Dare I say, kind of boring. We'll talk about the parts of the cell and what they do. But please stay awake. Don't fall asleep, because the second half of this chapter talks about diffusion, osmosis, active transport, which is talking about how we move things in and out of the cell. And that's really, I'm going to say it 2 more times, really important. So here we go. So as I said, the first half we'll talk about the parts of the cell. Then we're going to spend a lot of time talking about how we move things into and out of the cell through the plasma membrane. And then we probably have 1 or 2 slides, and that's it on cell division. Okay, as a reminder, why on earth are we talking about the cell? Didn't you cover this in 5th grade? Well, probably, but we have to review it because this semester we're going to be talking about every body system in the body. And guess what? Each body system is made up of organs, and those organs are made up of tissues, and those tissues are made up of, you guessed it, cells. And those cells, while they share some similarities, different cells have different things in them, and that matches the function of that type of cell. So it's really important that we can sometimes go to that cellular level and talk about, hey, look, this is a cardiac muscle cell. And you know what's really cool about it? It has these things called intercalated discs. And we're going to talk about why they're important. So in order to be able to do that, we have to be able to know about the cell, just the basic parts of the cell. All right, so as I've already said, it's like I planned it or something. I tell you, every cell has some of the same parts, but they can look very different. This stuff right now, you don't have to memorize. It's not going to be on a quiz or an exam. I'm just giving you an example. Look at how different a bone cell looks to a nerve cell to a red blood cell. So we'll talk about the basic structure, understanding that as we go through all the body systems later, we'll be able to identify some of their specialties. And of course, they're structured that way for a reason. Really cool, actually. Okay, here we go. I paused for a drink, I'm sorry. For each part of the cell, know its general structure and its general function. The location of each part of the cell isn't as important, but for every organelle, for every part of the cell, make sure you know its structure and its function. Here we go. Here is your basic animal cell. Isn't it pretty? You've seen this before? These are all the possible organelles that an animal cell, human cell could have. But not every cell will have all the parts. Yet some cells in our body will have most, if not all of these. Okay, but I'm going to go pretty quickly because it's very straightforward. Okay, know the basic structure and then the function. So, every human cell has an outer membrane that we call the plasma membrane. It is a specialized membrane. We don't call it a cell wall, that's only in plants. It's a plasma membrane. Inside the plasma membrane, we have tiny organs called organelles. So all of these bits and pieces, even the nucleus, is an organelle. It's a mini organ. And these organelles are, let me change a different color here. Why don't we do blue? These organelles are housed in a fluid called the cytoplasm. Any structure that we talk about, you'll need to know the basic structure and function. So plasma membrane is the outer membrane. Organelles are tiny organs inside the cell, and the cytoplasm is the fluid that these organelles sit in. You can imagine some very straightforward questions on our quiz, and then the midterm and the final. You know, the organelle, which of the following defines an organelle? And you would choose the answer that says tiny organ inside the cell. Now, we also have something called cytoskeleton. And this doesn't just mean one thing. It is a collection. So most of the time, people will refer to 3 different types of things that make up the cytoskeleton. You do not have to know these. I'm just pointing out that it's not just one thing. There's a variety of things that contribute to our cytoskeleton. And the cytoskeleton are, do you see these fibers? They're fibers that exist inside of the cytoplasm, and they just give the cell structure. As the name suggests, cytoskeleton, a skeleton gives our body structure. It helps give us structure. We don't want the cytoplasm to be too liquidy. We need this cell to be a little bit firmer. So we have these little protein filaments, collectively called the cytoskeleton, that give the cytoplasm a little bit of structure. These are the different types. Sometimes it's just more of a linear line. Sometimes it's a band of things or a round barrel structure. So these are examples, but you don't have to know these specific types of cytoskeleton. This picture I really like, and that's why I put it in here. You don't need to know them, but I like how it shows the cytoskeleton. So all these things going in between, this is more of a 3-dimensional view. Can you see how it acts like scaffolding? It holds things together. All right, moving on. The next part of the cell is really important, and I hope you remember some of these from your previous education. The mitochondria. Its structure is, it's kind of sausage-shaped, maybe peanut-shaped. Also importantly to its structure, it has an inner membrane. So in that way, it has a double membrane. And the importance of this is that it provides more surface area. We're going to see this trend over and over and over again. More surface area means that there's more of an area to be able to do whatever job it has to do. So the mitochondria, it is sausage-shaped, and it has an inner membrane. So collectively, we have the outer membrane, plasma membrane, plus we have the inner membrane, so we have a double membrane, which gives more surface area. The function of the mitochondria is, of course, to generate energy. A cell, it's like a little working factory. It's a living thing. And therefore, in order for all the other parts of the cell to do their job, it needs energy. Just like your car needs fuel, or a factory needs some kind of energy, whether it be from the workers, we need power, we need whatever. So the mitochondria is really important because it generates energy for the cell. There's some extra information on here, but you only need to know what it is I'm talking about. All right. The next organelle you should know are ribosomes. They're little dots. And we can see here some of the, it's kind of hard to see. Let me go here. Some of the dots are located on the rough endoplasmic reticulum. So all these little dots. As a matter of fact, this is called rough endoplasmic reticulum because it has these ribosomes. There may also be some free floating. So we also might see some out here. So some are attached to other structures, and some are free floating. The function of the function of the ribosomes is protein synthesis. Synthesis is the fancy word for production. So these little ribosome guys or gals, they make protein. Really, really, really important. I've been saying really, really a lot in this chapter. So the ribosomes, they make protein. Protein synthesis. Now, this is just to go back to chapter 2. Do you remember in chapter 2 we talked about the organic compounds, carbohydrates, fat, and protein? And remember, protein, we didn't need as much through our diet. I think I mentioned that. If not, it's true. But I know I mentioned we have 20 amino acids, the building blocks of protein. Some of them our body's able to make, which are called non-essential. And some of them our body cannot make, so we need to get them through food. But I'm just making a point. In chapter 2, we talked about how the body makes protein. Well, now I'm showing you the part. The ribosomes in pretty much every cell. Most every cell of your body has ribosomes, and they are protein-producing factories. See how it all ties together? All right. Next, we have the endoplasmic reticulum, or ER. It's a series of passageways. It's the blue thing, right? It usually, actually almost always, is located right beside the nucleus. There can be rough ER, which has the ribosomes. And then there can be smooth ER, which doesn't have attached ribosomes. And the smooth ER kind of looks more like coral reef. The function of these interconnected tubes, transport. It's all about transport. What we're going to find out shortly is that as those ribosomes make protein, we need to transport that protein. So it goes through the little interconnected tubes of the endoplasmic reticulum. Or if I were to draw it here, I'm not really sure where to start, but we could see that, maybe the little proteins travel through these tubules. So we have the endoplasmic reticulum, usually right outside the nucleus. We have rough and smooth, ER, and its function is to transport. See, folks, not so bad. Now, we also have another set of tubules called the Golgi apparatus. One of the best ways to identify this is to notice that the Golgi apparatus is not right outside the nucleus. Look at this picture here. We have the nucleus. And right outside the nucleus, we're going to find the ER. I just put it in blue. Well, separate from that, not attached to the nucleus, is another series of tubules called the Golgi apparatus. So it is not attached to the nucleus, but in structure, it's pretty similar. So it's kind of a series of passageways. And what the Golgi apparatus does, it organizes and sorts proteins. You might like the idea that it's kind of like a post office where it gets a bunch of things from everybody and the post office sorts it by address and then sends it out to the proper place. Well, we're going to find that the Golgi apparatus works in a similar way. What's going to happen? And we're going to go through this again, so you don't need to get it all down now. We have the nucleus. And around the nucleus, we have the endoplasmic reticulum. And of course, in the rough ER, we have these ribosomes. What we're going to do when we put it all together is we're going to follow protein. Because what we're going to find is, as the ribosomes produce that protein, the proteins travel through the ER, and then those proteins are released. Well, where do those proteins go next? Boom, you got it. They go to the Golgi apparatus. So those same proteins that just came from the ER are going to now make their way to the Golgi apparatus. And actually, that's what they're showing you down here. Look, there's a transport vesicle from the rough ER, and inside of that is protein. Once all the proteins get to the Golgi apparatus, and as those proteins travel through the Golgi apparatus, the proteins get sorted. And see, what happens is, well, some of those proteins we want to send to the outside of the cell. Maybe other proteins, like this one, we want to send to another organelle called the lysosome. Maybe even other proteins we want to send to the mitochondria. So you see, once the proteins get to the Golgi apparatus, they're organized, kind of like a post office. And then they are sent out to other parts of the cell. So the proteins that are sorted, they leave the Golgi apparatus and then travel to somewhere else in the cell. But because they've been organized, just like a piece of mail has an address on it, it knows where it has to go. Isn't that really cool? Well, as cool as a cell can get. This is a reminder that the endoplasmic reticulum is right outside the nucleus, but the Golgi apparatus is not attached to the nucleus. Okay, another example of an organelle I'd like you to know is called the lysosome. The lysosomes are round structures. And there might be one there might be one here. So lysosomes are round structures. And what they do is they break down bad stuff. I realize that this statement is a little general, but sometimes I find it most beneficial just to tell it like it is. So lysosomes, they break down bad stuff. Let's say that there is some old tissue. So maybe from other parts of the cell, there's an old organelle that's no longer functioning. Well, you know what? The cell is going to send that to the lysosome so it can be broken down. And then its contents will be either spit out or recycled. Maybe we have a bacteria over here. Well, that's bad. We got to get rid of that. Maybe we send bacteria to the lysosome. And the lysosomes will break it down, spit it out. And of course, different types of cells are going to have more or less of certain organelles. So I'm just looking ahead because trying to plant some seeds here. For example, lysosomes, because they break down bad stuff, they're more important in things like white blood cells. Okay, what is the job of a white blood cell? Well, it's part of the immune system, helps break down bacteria and bad stuff. So wouldn't a white blood cell want to have more organelles that break down bad stuff? Of course. All right, now, we can't forget about the nucleus. We know what the nucleus is. The nucleus is in the center of the cell. Here it is. What I want to point out about the nucleus is that it has a specialized outer membrane. Here, it's called the nuclear envelope. I'm not going to ask you that specific term, but if you just look at it, you'll see that indeed this outer layer is just a little bit specialized. It has 2 layers. The nucleus, it's really important. So anytime we have something that's really important, usually the body's going to put a more specific or tougher structure surrounding it as protection. So what is the function of the nucleus? You got it. Well, it's the control center. So if we need to send bacteria to the lysosome, if we need to tell the mitochondria to produce more energy, these directions are coming from the nucleus because it's the control center. And of course, the other thing about the nucleus is that it contains the genes. So we're going to have inside the nucleus our DNA. And that's pretty darn important. So again, another reason why we want to have this specialized membrane. Okay, cool. Nucleus, central part, has a specialized membrane, controls the operation of the cell, and it also contains our genes, DNA. Now, let me go ahead. Okay. When we talk about how, and I know you hear that word and you're like, oh, why can't we just keep it simple? Well, I really want to make one important point. When we talk about how, well, how does the cell tell the mitochondria to make more energy? How does the cell tell the cell to send the bacteria to the lysosome? Well, it's not like our overall body. You know, in our overall body, we have our brain, and the brain is going to oversee the overall body by sending out nerve impulses, which are electrical currents. Well, it's a little bit different in the cell. When we look at each individual cell, it's not like the cell, the nucleus sends out nerve impulses. Nope, that's not how it actually oversees the cell. Instead, the nucleus is able to oversee the cell by telling the cell what kinds of proteins to make. So at this cellular level, it is all about the proteins. All about the proteins. So when the ribosomes make those proteins, the nucleus, the control center, the nucleus is telling the ribosomes what kind of proteins to make. So it might make protein 1, or maybe it makes more of this other protein 2. Maybe it makes this other protein 3. That's not how they're named, but I'm just trying to make a point that these proteins are different. And maybe protein 1 is what goes to the mitochondria. So if the cell needs more energy, then the control center, the nucleus, will tell the ribosomes to make more of the proteins that go to the mitochondria. So the nucleus is able to oversee the cell by telling the cell what kind of proteins to make. And then, of course, those proteins go to the Golgi apparatus, which then go out to other parts of the cell and then do their job. And a lot of times these proteins leave the cell and go to other parts of the body. So it's not just that this controls the cell. As I already mentioned, some of those proteins, once they leave the Golgi apparatus, they go to the outer membrane and they leave the cell. And they can have actions otherwhere in the body. So it's all about the proteins. That's really cool. All right. This is we're getting towards the end of the structures here. There is a little dark structure called the nucleolus inside of the nucleus. And its job is to produce ribosomes. So the nucleolus produces ribosomes. Those ribosomes then go to the ER. And then what do those ribosomes do? Those ribosomes are little red dots. What do those ribosomes do? They produce protein. And those proteins travel through the endoplasmic reticulum, leave the ER, and then go to, I'm just going to keep going over it again and again, go to the Golgi apparatus, which organizes the proteins, and then the Golgi apparatus spits out the proteins which go to their ultimate destination. But it all starts with the nucleolus producing ribosomes, which will then go out to the ER. And of course, the ribosomes will go on to make proteins. All right, so I've already written this out, but I wanted to make sure that it was written on the slides one by one. But I've already said this. We'll just review it one last time. So the nucleolus produces the ribosomes. That's right here. The ribosomes travel through the nucleus. And the nucleus has the DNA. So as the ribosomes travel through the nucleus, they are told what to do by the DNA. So now these ribosomes are armed with an instruction manual. What kind of proteins to make and how many. Then those ribosomes travel into the endoplasmic reticulum. And what do the ribosomes do? They make proteins. So these ribosomes now make proteins based off of the instructions that they got from the DNA. Those proteins travel through the endoplasmic reticulum. Then those proteins, we know this, leave the endoplasmic reticulum and go to the Golgi apparatus. Those proteins are then sorted, and then those proteins are spit out by the Golgi apparatus to a variety of different destinations. This example, the proteins are going to a vesicle, which is going to spit them out of the cell. Over here, these proteins are going to a lysosome. These proteins are also going to a different part of the membrane to come out. Wow. It is like a little working factory. All right. You know, the exams, the online quizzes and the midterm and final, they're not essay, they're multiple choice or matching. So you don't have to be able to write an essay about those steps. But what if I give you a list and have you put them in order? So I do expect you to have an understanding of what happens in order. All right. Those are the only parts of the cell you need to know. There are more parts of the cell. Obviously, we didn't cover the centrosomes or something like that, the chromatin. I'm trying to simplify it. We have enough to know already. But what we discussed are the only parts of the cell you need to know. Okay, here we are, part 2. And this is important. Important. All right. We briefly mentioned that plasma membrane. Well, now we're going to talk a lot more about it. Now, why this is so darn important. That plasma membrane is so important because its job is to keep the good things into the cell and then push the bad things out. So it is the guardian at the gate. And that's why I have a picture of this doorman, perhaps, sitting outside the club. This doorman gets to decide who gets into the club and who doesn't. Ha ha ha. So it's really, really important, okay? It is structured in a specific way so that we can let some things in, but not all, and that we can effectively get things out of the cell that are dangerous. All right, let's first look at the structure. Now, in this first picture, this plasma membrane, you were seeing it from afar. What if we were to magnify that. When you look up close at that plasma membrane, it is made up of what we call a phospholipid bilayer. Each of these little guys kind of looks like a sperm, right? Has a head with the tail. Well, it's a phospholipid bilayer. Each of these is called a phospholipid. And as a matter of fact, you will recall we talked about phospholipids being a type of lipid in chapter 2, and that they were structural parts of the cell membrane. Well, now we're seeing them. They form a bilayer, a layer of 2. And the heads are on either end, and the tails face the middle. Really important. Phospholipid bilayer. Now, there are also, scattered throughout, some proteins. These are going to act as channels. If you look at the picture up top, we can see a cross-section, and you could imagine how this little protein could act as a channel. Okay, so the structure of the plasma membrane is a phospholipid bilayer. 2 layers of phospholipids, heads face the ends, tails face the middle, and then also scattered between the phospholipids are some proteins that, among other things, act as channels. Now, it's important to point out that the heads are hydrophilic, which means they like water. And the tails are hydrophobic. They don't like water. And this is important because this keeps water either here, near the head, or here, near the head. We don't want to get water stuck in no man's land. We don't want to get water stuck in no man's land. So it doesn't, because the tails repel water. They're hydrophobic. They don't like it. All right. Now, I've already mentioned the function. We're just going to give a special name to it, called selective permeability. That outer membrane allows some things in, and it allows some things out, to go out. So it is selectively permeable. You know, other things in our body are much more permeable. We're going to talk about areas of our kidneys. So if we look, if I can draw a kidney, that's not bad. And then here's the ureter coming out. If you look at the kidney, there's microscopic structures called glomeruli. And it kind of looks like a ball of capillaries up close. But the idea is that this glomerulus, if I were to look at the membrane up close, the glomerulus membrane has big, huge pores. So the glomerulus allows quite a lot to filter out of it. So it is very, very permeable. Well, of course, our plasma membrane is much more selective. So it's much more of a tight membrane. There are some very small pores that we can fit things through. Sometimes we have a little channel too to get things out or in, but it's much more selective. It allows some things in, some things out, but it is selective, just like the bouncer. It doesn't let just anybody in. Now, what are the reasons why this is so important? Well, as we've already established, the cell is a working factory. It's got a lot of things to do. It's got to make all these proteins. It's got to make the right amount, the right kind. It's got to send them to the right place. And all the while, it has to keep bacteria at bay. It's got to get rid of old tissue that's not working anymore. It's a full-time job. In order for these cells to do their job, they need some supplies. They need fuel. We bring in glucose that we convert to ATP. They need water, something like oxygen. And there's a bunch of other things as well. But in order for the cell to keep working as a little factory, it needs supplies. How do we bring those supplies in? We've got to transport them through the plasma membrane. And then Let's say that the lysosomes break down some bacteria. Well, how are we going to actually get rid of it? We got to get it out of the cell. And there's other things too that we have to get out of the cell. Carbon dioxide is another waste product that we must be able to move through the plasma membrane out of the cell. All right. Now, as we talk about ways in which we get things into and out of the cell, we can divide them into ways to move things that are passive, and this does not require energy. We can diffuse things from high to low concentration without any ATP energy. But then sometimes there's situations where we actually do require energy to push things. Sometimes it's because we're pushing things against a concentration gradient. Other times there's other reasons. But active means requires ATP. It requires energy. And I've listed the different mechanisms of transport that we're going to be talking about. Are you ready? We'll start with passive first. Okay. And I apologize, when I move things to my iPad, sometimes the arrows and things don't work. So for example, this is supposed to point to an area of higher concentration. This is supposed to point to an area of lower concentration. The arrows don't show up. All right, so make sure you can define diffusion. And of course, diffusion is movement of particles from a high to low concentration. Movement of particles, molecules, whatevs, from high to low. This happens in the body. It happens in real life. You're in the kitchen baking brownies. Doesn't the smell of the brownies make its way to the living room? That's diffusion. Those little yummy smell particles are diffusing from areas of high concentration in the stove to areas of low concentration in the living room. Same thing here. So for example, here we have a lot of green particles. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we have we have 11 green particles on this side of the membrane. Here we only have 1, 2, 3, 4, 5, 6. Here we only have 6 green. So guess what? If we have a semi-permeable membrane, which we do, we're able to diffuse from areas of high concentration to areas of low concentration. Sometimes we diffuse through a channel. Other times we can diffuse right through, if it's a small particle, sometimes we can diffuse right through the phospholipids. Okay, what is required for diffusion? We have 2 requirements for diffusion. There must be a concentration gradient. One side must have higher amounts than the other. And the other thing is that the area that you're trying to move it through must be at least a semi-permeable membrane. Of course, if these phospholipids were not at all permeable, we wouldn't be able to diffuse. Cool. Okay, this is what I was saying before. Some things diffuse through a protein channel. Other things can diffuse right through the phospholipids. Either way, it is passive, moving from high to low. I've already said this. I tell you, we're so good. There has to be a concentration gradient, and there has to be at least a semi-permeable membrane. Ladies and gentlemen, I know I get excited. I wish you could see me. But I can't figure out how you can see me, and I can also draw on the screen at the same time. I get so excited. You know, the reason why we talk about all this, we're not just trying to be boring science people, because diffusion is a mechanism that our body utilizes. When we get to the respiratory system, do you know how the body exchanges gases in the lungs? From the lungs to the surrounding blood vessels? Diffusion. So the body is set up that way. the alveoli of the lungs, this outer membrane, is semi-permeable. So when we breathe in, we have a high amount of oxygen in the lungs, in the alveoli. And we have much lower amounts of oxygen in the bloodstream. So because the membrane is semi-permeable, and because we have higher amounts of oxygen on one side compared to the other, boom, diffusion. So your body is smart. It doesn't want to work harder than it has to. So it sets things up so we can rely on the natural process of diffusion. It's passive. We don't need energy. We're not wasting energy to try and do that. It's lovely. Another example is how we absorb nutrients from the inner walls of our GI tract, our small intestine, into our bloodstream. Guess what? Diffusion. All right. Another way that we can define this is osmosis. And really what osmosis is it is diffusion of water. When you hear osmosis, you automatically want to think about water. When we say just diffusion, like we just talked about, we want to think about particles, oxygen, glucose, potassium ions, nutrients. So when you see or hear diffusion, we're thinking of particles, aside from water, moving from high to low concentration across a semi-permeable membrane. When we say the word osmosis, we're specifically referring to water. It's the same principle. Water moves from high to low concentration passively. It's got to have a semi-permeable membrane. So here on this side, we have more water. 1, 2, 3. Here we have less water. There's no water. So how is water going to move? From high to low concentration. Sometimes water will move right through the phospholipid heads. Other times water will move through a channel. Just depends. But when we say osmosis, it is simply diffusion of water. And we diffuse a lot of water. Just like we diffuse particles, we also have to be moving water. All right, again, I'm just repeating myself. Some water moves through a protein channel. Other water can move right through the phospholipid bilayer. Okay. Now, I'm taking a breath because I want you to take a breath. There's a couple concepts that we've got to fill in here, and they're a little harder. So let's just get our bearings here. We're talking about mechanisms to move things into and out of the cell, right? Through the plasma membrane. And we've completed our discussion on how we move things through the plasma membrane passively. We can rely on diffusion and osmosis to move things into and out of the cell through the plasma membrane from high to low concentration passively through a semi-permeable membrane. Now, we're going to pause and talk about these concepts. When we come back after this, we're going to pick up our discussion and then talk about the active ways to move things into and out of a cell, the things that require ATP, like active transport, like endocytosis and exocytosis. But for now, we're taking a detour because we need to talk about osmolarity and tonicity. Aren't you excited? Okay. Now, osmolarity has a lot of different definitions. The definition that I'm using is pretty simple, still correct, but it is really tailored to what we're talking about. So the definition I like to use for osmolarity is the concentration of a solute in a solution. And I like to use good old-fashioned Kool-Aid. Let's break these things down. So, first of all, in a solution, whether it's Kool-Aid, in the body, whether it's the blood, okay? Any solution has two parts. There's the solute and the solvent. Do I have a slide on this? Okay. The solute is whatever is present in smaller amounts versus the solvent. The solvent is whatever is present in larger amounts. Well, I'm going to tell you, in the world and in our body, a lot of times the universal solvent is water, especially in our body. We have a lot of water, and it's for a reason. It's such a good solvent. Things can dissolve easily into it. But by and large, the body's universal solvent in any solute, any type of fluid, is water, because there's more of it. So I'm going to stick with this Kool-Aid analogy. Well, what are the two ingredients in Kool-Aid? You could also use Gatorade as an example. What are the two components? Well, we have the sugar, the powdered sugar, or the powdered Gatorade sugar, and we have water. Now, of course, there's going to be more water than sugar. So we make water the solvent, and we make sugar the solute. So, of course, we're going to have sugar, but that sugar is dissolved in a whole bunch of water. Now, even if you like a really sweet Kool-Aid, you're still going to have more water, okay? So the osmolarity is the concentration of the solute. It's not the amount of the solute, because we have to talk about the amount of the solute relative to the solvent. So that's why we use the word concentration. And I'm going to give you an example in a minute. So now that we've defined the terms solute and solvent, we can more easily understand, okay, osmolarity is the concentration of the solute. The concentration of the solute. Okay, let's make two different Kool-Aids. We have one Kool-Aid down below. Let's make another one. Here's I'm a really terrible drawer. How am I doing? There's a reason why I don't teach art. Okay, so let's make a high osmolarity Kool-Aid. So in a high osmolarity Kool-Aid, we're going to add a lot of sugar. So it's high osmolarity because it contains a high amount of solute. What is that Kool-Aid going to taste like? If we have some water, but we have a lot of sugar. Let's say I have 90 parts water and I have 10 parts sugar. What is the percentage of sugar? 10 out of 100 total parts. So it's 10% sugar. That's a lot, folks. If you think about, that's a lot of sugar. But it tastes very sweet. So a high osmolarity solution has a lot of solute. And think of it tasting sweet. On the other hand, a low osmolarity Kool-Aid contains a lower amount of sugar. It would taste more watery. So we still have water, but we just have less sugar. Maybe we have only 5 parts sugar, which means the remaining 95 parts is water. So in this solution, it is 5% sugar, and it will taste more watery than the other one. So osmolarity is the concentration of the solute. So I'm going to give an example in our body, and we're going to talk about the bloodstream. So let's talk about a normal bloodstream. Here's the blood, normal. And then down here, we'll talk about a dehydrated bloodstream. So let's first identify the solute and the solvent. Well, the solvent, as it is almost always in the body, is water. And the solutes are blood cells. So just to let you know, normally the blood has a very high osmolarity because about 55% of the bloodstream is the water or plasma, and 45% are solutes. Can you imagine a 45% sugar Kool-Aid? It would probably have the consistency of like quicksand. It would just be mud. It would be so much sugar. But we want the bloodstream to have a high osmolarity. We want to have a lot of blood cells. So these are blood cells. The more blood cells we have, the better we can transport oxygen, the better we can fight disease. We want to have a lot of platelets, right? And then those blood cells and platelets are immersed in water, the solvent, which acts as its fluid medium. Okay, so here we go. Normal blood is 45% cells and 55% plasma. Now, as we talked about in our previous example, one way to alter osmolarity is to increase or decrease the solute. So in our first example, we increased the solute. We added more solute. The water stayed the same, but we added more sugar, so it made it taste sweeter, higher osmolarity. In this example, we lowered the solute. We put less sugar into the same amount of water, which means it was a lower osmolarity and it tasted more watery. So one way to alter osmolarity is to increase or decrease the amount of solute. But another way is to alter the amount of solvent. Not only can we alter the solute, but we can alter the solvent. Think about when you are dehydrated. When you're dehydrated, we're still going to have the same, we're very close to, amounts of cells, trying to draw out the same as above here, and platelets and other goodies. So we still have the same amount of solutes. But what we've done, when you're dehydrated, we have less water. So instead of there being 55% of water, maybe we only have 50% of water. Okay, so we've taken out some water, making the relative contribution of the cells more. So now the cells make up 50% of the blood, and the plasma makes up only 50%. Well, what happens to the osmolarity? The osmolarity is the concentration of the solute. Here, the solute is 45%. Here, the solute is 50%. So even though we haven't altered the amount of solute, by decreasing how much water there is, the relative concentration of the solute goes up. So when someone is dehydrated, the osmolarity of their blood goes up. Not because we added more cells, but because we took away some of the water. And that can be bad. You know why? Because it makes the bloodstream too thick. So just imagine, instead of the heart trying to pump water, mostly water, through your blood vessels, now the heart has to pump something much thicker. So imagine the heart trying to now pump out maple syrup. Isn't that denser? So it puts more work on the heart. And because of that, it's not able to pump out as much of the cells, and it can be very problematic. I tell you what, isn't this good stuff? Okay, stay with me here. Another important concept is tonicity. Okay, now these are the toughest parts of this chapter. The rest of this is not so bad, I promise. So stay with me for this, and then we're going to coast to the finish. And the good thing about this video is that you can watch it over and over again. Okay, if you really are dedicated, you can do that, and then you can reach out to me if you have questions. You can email me, you can request a Zoom call, and I can answer your questions or explain it in a different way to hopefully help you. All right, here we go. Tonicity. You are going to see tonicity probably defined differently in a textbook, but I much prefer this definition. How a cell may change shape. That's the key. I mean, let's not complicate it. We have a cell. The cell membrane has selective permeability. Sometimes things diffuse in, sometimes things diffuse out. Well, depending on the situation, this is going to change. So what happens if all of a sudden a lot of water comes diffusing in? Well, anytime a lot of water comes diffusing in, the cell is probably going to get a little bigger because we've just pumped a whole bunch of water into it. So how much we diffuse into and out of the cell, of course, can lead to the cell getting bigger or smaller. That's all we're saying. Okay? That's all this concept is saying. Because of what's diffusing into and out of the cell, it's possible that sometimes the cell may get bigger or smaller. That principle, ladies and gentlemen, is tonicity. Now, this is a pretty classic way to define it, and we're talking about it because it can happen in the body, and it can be very dangerous. I just mentioned dehydration, how it will change the osmolarity of the blood. Well, if that gets to be too bad, the cells are also going to change shape. And if the cells change shape too much, they can't do their job. And once cells can no longer do their job, we are in trouble. So, we're going to lay out 3 scenarios. We have a beaker, and in beaker 1, we have red blood cells in a salt solution. So in beaker 1, the red blood cells are in mostly water with some salt. In beaker 2, we're putting those red blood cells in 100% water. So it's just water. In sample 3, we're putting the red blood cells in an even higher salt concentration. This is about the equivalent of seawater. So here, we have the red blood cells immersed in a much saltier solution. So the question is, in which scenario does the cell get bigger? In which scenario does the cell get smaller? And in which scenario does the cell stay the same size? Well, the pictures are showing you what's going to happen. Okay? In the first situation, the red blood cells stay the same size. We like that. In the second example, the red blood cells get bigger. And you know what could happen? They could burst, and that's really bad. And then in the third example, they get smaller. They crinkle, and then they can't do their job, and that could be just as bad. But the question is, why? I know. Why are we talking about this? Okay. I have these here, but what I'm going to do is I'm going to open up a new one because I want to draw. Let's see. New one. I know, call me old-fashioned here. Create. Look at me using technology. All right. So what we're going to do is we're going to draw our beakers. That was A, this is B, and this is C. And I can go back here, which is really cool. So in beaker A, we had the red blood cells put into a saline or 0.9%. So we have mostly water, actually in all of them. We have these red blood cells in all of them. But in the first scenario, it is 0.9% salt. And by the way, if this solution is 0.9% salt, by process of elimination, if it's salt and water, and 0.9% of it is salt, how much of it is water? 99.1%. We just know that it's a saline, right? Saline is a salt water solution, salt and water. So if it's 0.9% salt, then we automatically know it's 99.1% water. In the second scenario, we put the red blood cells in 100% water. So this is 100% water. And then in a third example, we had some water, but we had a 3.5% salt solution. And again, same thing happens. If we know it's a salt water solution, and if 3.5% of it is salt, then the remaining part of it, 96.5%, is water. So how do we get to the point where we figure out in which scenario do the cells stay the same, get smaller, or get bigger? And the answer is simply going to be we compare. We're going to compare what's the fluid in the red blood cells to what is the fluid outside of it, this fluid. So we're comparing this and this. By comparing, we can just figure out where water and where the solutes are going to flow, because guess what? We know that red blood cells have a semipermeable membrane, and we know that if you have a semipermeable membrane and you have a concentration gradient where something is higher on one side than the other, what's going to happen? Diffusion. So we've already outlined the composition of the beaker water, right? So on this side, I've put the composition of the beaker. And the beaker water is what this is, right? So now let's talk about what is the composition of the red blood cells. Because there's fluid in there. We already know there's cytosol, there's fluid. Well, this is just a known. So this is just something that we know to be true. And in every case, red blood cells, the fluid inside of red blood cells is just like saline. So we just know for every red blood cell, the fluid inside of it is 0.9. Let me use black, be easier to see. Because the inside of the red blood cell is a saline solution, we just know it's 0.9% salt, which means it's 99.1% water. Same thing here. These are the same red blood cells, 0.9% salt, 99.1% water. And then, of course, here it's the same thing, 0.9% salt, 99.1% water. So I'm not going to ask you to replicate this in an essay, but I'm purposely trying to walk you through it because I want to make a point that is this. When we are trying to determine what's going to diffuse and in what direction, if we're trying to determine what's diffusing and from where to where, all we have to do is compare the fluids on either side of the cell membrane. If we're looking to see what's going to diffuse and where, we just look and see, well, what's on either side of the cell membrane. Here we're talking about red blood cells. So let's take example number 1. If we look at what is inside of the cells, 0.9% sodium, 99.1% water, and we compare it to what is outside of the cells in the beaker water, they are the same. So if the concentrations of water in the solute are the same, there's no movement. Because remember, in order for there to be diffusion, we have to have a concentration gradient. So here, cells stay the same size. And that's what we saw in our first example. Here, the cells stayed the same size. And why? Because the concentration inside the cells of fluid and the concentration outside of the fluid, outside of the cells, is the same. This, ladies and gentlemen, this type of solution is called an isotonic solution. Iso tells us something about the same. So in an isotonic solution, the fluid compositions inside the cell and outside the cell are the same. So there's no diffusion of those particular things, which means the cells stay the same size. Okay, next example. Here we still have 0.9% sodium and 99.1% water. But now, outside of the cell, we have 100% water, which is 0% salt. Now, when talking about diffusion, we're going to want to focus on the water because there is always more of it. So if a cell is going to change shape, it will be because of the movement of water, because there's so much of it. Let's look at water. Here we have 99.1% inside the cell. Here we have 100%. So how is water going to move? It's going to move from high to low. Water is going to move from higher concentration here to lower concentration here. Water diffuses into cells. As a result, cells get bigger. And that is what happened in this example. Water diffused into the cell and the cells get bigger. This type of solution is called a hypotonic solution. Now, it gets a little confusing because just as water is diffusing into the cell, sodium is also going to diffuse. Inside the cell, there's 0.9% sodium. Outside, there's 0. So sodium will diffuse from higher amounts inside the cell to lower amounts outside the cell. And that's where the name hypo comes from, because there's less salt in the solution compared to the cell. But I think that's confusing. Even though the solutes are diffusing, there's just not that much of it. Always look at the water, because it is the movement of water that's going to change the shape of the cell, because there's more of it. And then, of course, we have our last example. You should be old hat at this by now. We're going to compare the water. Inside the cell, we have 99.1% water. Outside the cell, we have 96.5%. So we have more water inside the cell. So water will diffuse from higher concentration inside the cell to lower concentration outside of the cell. Water diffuses out of the cell. And as a result, if the cells are losing water, cells get smaller. This type of solution is called a hypertonic solution, when the cells get smaller. And that's what's happening over here. Water diffuses out of the cell, cells get smaller. Okay, you do not need to repeat this exact explanation. But what I would like you to know is the overall point. And I'll say it again. When we want to know how something is going to diffuse, we just compare the concentrations inside the cell and outside the cell. You should also be able to define hyper, hypo, and isotonic. I don't have a summary slide here, but just be able to know that in an isotonic, what other color can I use here? how about green? In an isotonic solution, cells stay the same size. In a hypotonic solution, cells get bigger. And in a hypertonic solution, cells get smaller. How I remember it, and this might sound stupid, maybe it works for you, maybe it doesn't. I always think hyper. If someone is hyper, they may go see a shrink. So hypertonic, the cells shrink. Sometimes whatever little tool works for you, go with it. And by the way, this is why the IV bags have to be isotonic to the body. When we want to rehydrate just the plasma of someone's blood, we must give a solution that is hypotonic. I'm sorry, it's been a long day. Isotonic. Let me say that again. Burp, 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 3, 2, 1. When you give an IV bag to rehydrate the plasma, that IV bag must be isotonic to the blood, meaning that isotonic, there's no change to the cells. Now, there are some scenarios where you may wish to purposely give a hypotonic cell to purposely give some solution to the inside, but by and large, a regular IV bag, isotonic. You should know that. Okay, now, coming down to the end here. Thanks for sticking around, or maybe you did it in a couple of parts, whatevs. So now we're coming back to how we move things across the membrane. We can move things through diffusion and osmosis, which are passive means, but every once in a while, we have to rely on an active way to move something into and out of a cell. Here's how we do it. One way is called active transport. No one should miss that. It has the word active in it. Active transport requires ATP. In this example, we're looking at orange diamonds. This side, we have 1, 2 orange diamonds. This side, we have 1, 2, 3, 4. Well, sometimes you actually want to move things against the concentration gradient. So instead of using diffusion from high to low, which is passive, sometimes you really want to move these orange things from low to high. Listen, folks, if you're going against the concentration gradient, you will absolutely need ATP. Just like trying to row upstream. If I'm going downstream from high to low current or pressure, it's passive. But if I want to go against the current, I need some energy. And that's exactly what active transport is. We need energy because we're trying to move something from low to high. Good. Another way that requires energy is something called vesicular transport, which just means that we're transporting something, here's a something, but we're transporting it through the cell via a vesicle. Vesicular transport, using a vesicle. We have two ways, endocytosis and exocytosis. Whenever you want to use a vesicle to move something into a cell, it is called endocytosis, within. Whenever I want to use a vesicle to move something out of the cell, it's called exocytosis. I think about exiting, leaving. This is just straight memorization, folks. Okay, good. So the last thing for this chapter is cell division. We are not going to spend a whole lot of time. You can start your watch now. I bet you it's going to be 2 minutes. Mitosis and meiosis. We need to have an ability for our cells to reproduce. We are not born with the same number of cells that we die with. We need to be able to regenerate, fix damaged tissue, etc., etc., etc. There's 2 different ways to divide cells. And I apologize for this being a little bit, the formatting being off. Mitosis, and I've put in bold what you need to know. Mitosis is how most cells divide and reproduce. Meiosis is the term for when we do cell division, cell reproduction in sex cells only. This would be the sperm cells and the egg cells. That's it. Every other cell utilizes mitosis. Structurally, there's only one set of divisions. Now, I don't like the word phase here. Maybe I can add an S to it. Because there's actually 4 phases: prophase, metaphase, anaphase, telophase. So maybe we can say there's just one set of phases required. You only have to go through prophase, metaphase, anaphase, telophase once. Whereas in meiosis, you have to go through that same set twice. Takes longer. They're similar phases, you just have to go through them twice. In mitosis, you get from a parent cell 2 daughter cells that are genetically identical. You have one parent cell, and you produce 2 daughter cells that are identical. Whereas in meiosis, you have 4 cell yield. So we have one parent cell. We go through 2 sets of divisions. I'm messing this up. Hold on the phone. Here we go. 1, 2, 3, 4. But the resulting 4 cells are not identical. Think about a mother having 4 kids. The kids may share some things, but they're not identical. So look, I'm trying to show some differences. This first one is a half circle filled in. The second one is a full circle filled in. The third one is a triangle. The fourth one is a square. That's all you need to know. Okay, that's the end, folks. I'll see you next time.